So let us start.
Okay, thank you, sir, for giving me this opportunity. It's an honor actually to be able to present in front of such esteemed people, everyone who is much senior to me. Most of them are faculty, so it's an honor here to be part of this whole thing and present here. So, as sir has already explained much about homing endonucleases, I'll be touching some part of it which has a structural basis and understanding it from a computational point of view. So, just giving a recap of whatever Sir said, just highlighting the main points out of it. So, homing endonucleases are encoded by mobile genetic elements, as he already said. These can move from one place to another. So, which propagate by a process called homing, right? The endonucleases cleaves a cognate allele from which the mobile element is absent. Thereby stimulating a homologous recombination event that duplicates the mobile DNA into the recipient's locus. That's what Sir just said. They will cut their own they'll cut a sequence and send their own gene inside that. So the generation of engineered meganucleases with tailored specificities has become a field of research interest. This is your face is dark. I am dark, but in dark. I think because of the lights. I'm sorry about my color. So anyway, what we are trying to discuss here is uh, the thing is the, yeah, I got it. I got it. So the thing is, as sir said, the specificity of these meganucleases are very high, right? Because the sequence here is very long. So whenever we have a long sequence, the specificity will increase, right? Because but I mean, mathematically speaking, just to get one nucleotide space, suppose we want to predict something, if it's an A or it's a T, G or C. So the for one place, suppose the probability of that happening will be 1/4, right? That place can be filled by either A, T, G or C. Suppose we get an A. So A divided by, 1 divided by 4, that is 1/4. So every time we are trying to predict something, it will be multiplied 1/4 into 1/4 like that. So to the power n will be the sequence length. So suppose we have a sequence of four, like GAATTC, GAATTC, so six. So right. So suppose we are trying to find GAATTC, which is of 6 nucleotide length. So that will be like 1/4 to the power 6 probability of finding it, because the length is 6. Suppose we have something which is of 10, it will be increased because the power will be increased to the power 10, right? So in these cases, meganucleases, it's very long, right? 22. So to the power 22 is way too much, and it's very hard for a genome, I mean, an organism to possess such sequence again and again. So the thing is here, what people try to do is change the recognition sequence. I mean, the target, the meganucleases, the way it goes and binds, they're trying to change the recognition site so that it can be used to target more than what it naturally does, right? So we can artificially make the meganucleus to go and cleave the areas that we want to. So that is a part of the research. I'm sorry, just a second, someone is asking something. Can you please show a snapshot of any programming language that you are using to understand the breakage? Okay. What kind of breakage are you talking about? So anyway, I'm going to talk about breakages. I am just going to show you how to search for a given target, right? If you're given a target like GAATTC, suppose you want to search it in your database, what will you do? Of course, you'll just control F find and GAATTC. But the problem of using control F is it's very strict, right? Whatever you type will be exactly matched. So if you have something like Tata box, you cannot search control F using a Tata box, because the TA length might is variable. There is no specific TATA sequence. So that's why we need something flexible to search for such signatures, and that's what we'll be discussing more. So if you want to know about the programming language, I'm not concentrating much about the programming language here, but what we'll be concentrating more about is regex. It's called regular expression. So that's what we'll be concentrating. So it can be implemented in any language. You can use Perl, Python, or anything. So programming language is not a big thing here. So but I'll be showing it in Python. So I'm more comfortable with Python, so I'll be using Python. And moreover, if you want, there is no snapshot exactly. So what I'll be doing is, if you're familiar with Python, then I'll be just using a Flask to give a structure, nothing more. Flask is a module which gives a structure, input and output, it will give you a structure to it. So anyway, moving on. Oh, this camera is not good, right? Is it fine? Okay. Okay. Such proteins could be used to cleave endogenous chromosomal sequence and lead to wide range of application. Yes, Python would be good. Okay, thank you for agreeing with me. So anyway, moving on. On the basis of the primary sequence homology, four homing enzyme families have been identified, as was discussed by Sir. These are the five families. Laglidage, GIY, YIG, HNS, HNH, and histine, cysteine box. So out of this, Laglidage is the most, largest family. It contains most of the homonucleases. So largest family, Laglidage, contains several hundred identified members, many of which have been shown to be functional endonucleases, right? So uh, am I audible to everyone? Someone just said I think it's working, right? Yeah, it's working. It's working. So yeah, it's fine. So whenever we are talking about homing nucleus which belongs to Laglidage, so in that case, there are two monomers, right? So they form a homodimer. So what happens in one monomer? It has alpha, beta, beta, alpha, beta, beta, alpha, like that. On the motif, right? So as Sir said, it forms a saddle. Saddle is like what we put on a horse, that gives a structure. So we ride on it. So that thing which looks like a you know that gives a structure is called a saddle. So what happens? This beta sheets actually goes and binds to that DNA in between, and that's how it stabilizes. We'll see the structure in the next slide. So the DNA binding depends on the four beta strands, of course, from each domain, folded into an anti-parallel beta sheet and forming a saddle on the DNA helix major group, right? So major group, minor group, I think a lot of discussion was done yesterday. So I need not talk about it anymore. So this is what CRE looks like, right? So if you can see over here, like alpha, beta, beta, alpha, can you see this thing where I'm moving the cursor? I hope you can see it, right? So the sequences, yes, okay, anyway. As you can see, the first subunit is alpha, right? The alpha one, this alpha one. Then it is linked to beta one, right? The beta one is here, this beta one. And beta 1 is connected to beta 2 with a beta loop, beta turn, right? So beta turn will give this turn, for which beta 2 is there. So as you can see, this gives a net like structure that will sit on the DNA, and that's how it forms like a saddle, right? So if you see the whole structure over here, it is given as alpha, beta, beta 1, beta 2, then alpha. Yes, alpha, beta 1, beta 2, then alpha, then again alpha, beta 1, beta 2, and alpha. This is one monomer. The same thing happens here in the other monomer, right? So this thing will actually come and bind to this place, right? So if you look over here, there is minus 12 to plus 12, right? And in between you will see plus 1, and here there is minus 1. So this is the actually recognition site. The recognition site contains four base pairs, four meganucleuses. So these four are always common. So GTAC is the central for nucleotide for CVUI, and CREI has central CATG, right? I think. Okay, anyway, we'll be moving on and we'll confirm it anyway. So that is the thing. The cleaving is done in minus 1 and plus 1. As you can see, there where the cleaving is done, right? I mean, just here. The cleaving will be done here. So this part and this part, right? There will be a double strand breakage. So this is how the whole thing works. It will come, it will sit on it. The beta strands will work like a saddle, which will bind to the whole DNA thing. And there are two dots, if you see here, right? These are the calcium ions. This it works like a catalyst. So in presence of that, the whole thing will be catalyzed, and the double strand will be anyway. So moving on, let's talk about the engineering part of it. Since we know these are the naturally occurring homonucleases and these are the targets, but if we want to engineer it so that it can go and bind in the places that we want to, some customized place, so for that a lot of research has been going on. So although a large number of homing endonucleases have been identified, the landscape of possible target sequence is very limited limited to cover the complexity of the whole eukaryotic genome, of course, because yeah, sure. So the thing is, where was I? Where was I? Yeah. So as I said, the specificity is too much. I mean, 22 length is a very huge sequence to have, a specific place, the same space, same sequence signature in many places of a genome. That may not happen, or it's very, it's the occurrence is very less. As I said, the reason is 1 by 4 to the power 22. That is the probability, and which is very, very less. So that is why what scientists are trying to do is they are trying to redesign the whole thing. So this limitation may be overcome by redesigning natural homing nucleases to recognize novel DNA sequences, right? And suppose the homing nucleus, which does not actually target DNA sequence because it doesn't have that specificity, somehow you change the nuclease, the structures by alternating the amino acid residues, and now it is able to go and recognize that DNA, sequence which you want it to target. Anyway, so these redesigned homing endonuclease variants must induce a precise DSB near the targeted mutation to be suitable for therapeutic application. Of course, that's what the whole point is, right? There should be. I mean, it's better if we do the DSB to prevent some disease. Just doing random DNA cleaving is not helpful. So people are actually trying to find some gene like RAG1 gene. We'll be talking about it. So they try to correct these genes by making the DSPs. Okay. So let's see the whole thing, how the engineering is done. Though the first thing which was proposed was punctual redesign HEs, right? So here what they do, they just change one amino acid residue, and that will result in the change in the recognition site. As you can see over here, CRE1 in wild type actually goes and binds to A in the 3 position, right? But after they modified the target here, right, they modified the nuclease by changing one amino acid. Now they are able to target this T over here and an A over here, right? There are small changes, right? So this was the first thing that was proposed. Then what happened? They tried to target a whole gene. For that, they changed not only one amino acid residue but couple of them, so that it goes and binds to that specific DNA. So like that, as you can see, in this sequence, in the modified sequence, there are a lot of sequences given in yellow, right? These are actually the modified, I mean, the sequence which was newly added, or the nuclease enzyme which is now able to target these sequences. In wild type CRE1, they can only target this thing, CAAACGTC, right? But after engineering, now they can target TGTTCTCAG. So like that, they are changing some of the amino acid residues so that the nuclease enzyme goes and binds to new targets. That's the whole point of the engineering part, right? So people are actually trying a lot of things. They are sometimes they are changing one amino acid residue. Sometimes they are changing a cluster of amino acid residues. So as you can see over here, this yellow part is the changed amino acid residue, right? This yellow part over here, this one mutation in one amino acid mutation. Here you can see a bunch of amino acids that has been altered. This blue part and this green part, right? They change some quite an amount of amino acids. So in the same way here as well. So moving on, there is another approach where what they do, they take two mm, uh, meganucleases and they combine, right? So the central four are same. From the left, they will take one endonuclease, one monomer, and for the other, they will take another subunit. Not monomer, subunit. It's better to call a subunit because we'll see why. So let's start from the beginning again. Just understand what we were talking about. The first attempt, as I said, the initial attempt to redesign the DNA binding specificities of these enzymes were performed on a small scale by mutating single amino acid residues interacting with the DNA base, right? So only one amino acid residue will be changed here as it is shown, and it will change. So as you can see here, this is CREI, right? So here the recognition site is GCACT, right? Or on the other side, let's talk about this one, GTGA. That's better. So GTGA is the four nucleotide which are the central nucleotide where from where the cleaving is done. So after they engineered it, you can see the CAA is changed to CTAA, right? This one change over here. Then there is another change over here. This CA has become CG, right? The remaining are same. The cleaving is done in the same place, but they have just altered one base, right? The same way, if you look over here, it is CNG over here, and here it's ANT, right? It's C and it's A. The same way here, it's G, right? They have changed an amino acid residues for an amino acid residue for which now this enzyme is able to target the DNA sequence which is not originally, it is known for the binding study, the naturally occurring sites. So that's how the engineering part is done in one base substitution. The next substitution, as I said, is they take a gene and try to change the endonuclease accordingly so that it finally goes and binds to it. That is one of the other methods. So fully redesigned HEs were generated using ICREI scaffold, and these variants could recognize and cleave new target sequences contained in specific gene, right? So this thing was done in 2010, and they published this paper here, molecular basis of engineered meganucleases targeting of the endogenous human RAG1 locus. So RAG1 is actually in mutation in RAG1 produces a disease called SCID. So anyway, moving forward, the modification in homodimeric ICRI endonuclease through protein engineering to target a specific DNA sequence within the human RAG1 gene. So as I said, RAG1 is actually responsible for mutations in RAG1 is responsible for the cause of SCID disease. So they reported the structure of two engineered heterodimeric variants and one single chain variant of ICRE1. So as you can see in this color in magenta over here, these are the changed amino acid sequences, right? In yellow here. So these were the changed amino acid sequences. In complex with the 24 base pair oligonucleotide of the human RG1 gene sequence. Show the DNA binding is achieved through interactions in the major group, right? Most of these actually goes and binds to the major group itself. There are very few. So anyway, moving forward, as I said, the last one of the flow chart that I showed, that they take two different subunits and make a chimera, right? So another approach to amplify the number of DNA sequences recognized to generate novel chimeric homing endonucleases using the subunit identified as structural blocks in a domain swapping procedure. They swap the domains, right? So DMOCRE is a redesigned chimeric variant that was produced based on DMOI and CREI. So DMOI is one endonuclease and CREI is another nuclease. So what they did, I'll show you over here. Where is my cursor? Okay. So this is the sequence of CREI, right? So if you see, this is the sequence here, and DMOI is the sequence given below. Now the variant, the chimer, which targets the sequence is given here, where the right part of the sequence given in yellow, you can see it is derived from ICRE, right? This, the target sequence has changed to this, and the first part, this in blue, belongs to this DMOI. So when we actually add this to CREI plus GM1, we make a timer of it. Now it is able to target and completely is and half CREI. So this is how the engineering part is done. So anyway, as you can see, there are a lot of sequence structure and everything is involved here. So of course, bioinformatics is going to come into the picture to help the biologists who are actually working on this field. So through bioinformatics, what we can do is we first keep a track of whatever the homoendonucleases are from their sequences and make some conti- uh, contigs, or what you call it, consensus sequences, right? So if you make some consensus sequences, we'll talk about it in the upcoming slides. So that's how you can go for more varieties of endonucleases, HEs. So let's start. For this was the first bioinformatic work that was reported. So in this work, they said, uh, I'm sorry, why is it called punctual and full? Because in fully they are particularly targeting a gene, okay, like RAG1. That's why they are calling it fully. But in punctual, they are just changing one nucleotide, and that might not be a part of a gene. They are just testing it, right? So here they have changed few couple of amino acids so that it can go and bind to a completely new gene. That's why it's called fully redesigned, right? Okay, thank you. So moving on. Are there any more questions? Because now I'll be going into bioinformatics. I'm stopping the biology part here. Anyway, so from this paper, what happened? Many hundreds of recognizable LH genes can be found within microbial genomic sequence. They said these things in this paper. Okay, so a significant fraction of these genes encode active endonucleases, right? Their target sites can often be determined by a combination of comparative genomic analysis and biochemical experimentation. Even closely related LHE homologs can display considerable diverse DNA cleavage specificities. Of course, this is understandable from what we saw in the engineering part, right? We are just changing one or two amino acid and everything has changed. So this led to the need for creation of database, right? That has homing endonucleases in it. So predictability of homoendonucleases, a target recognition, may allow the discovery of formerly unidentified HESs targets in the human and other genomes. That's what they said for the benefit of for the benefit of gene therapy and genetic engineering, right? So this was the need. So for which these researchers came up with this database, and they published in 2011. Native homing endonucleases can target conserved genes in human and in animal models, right? So while they did this work, they a huge amount of data was accumulated, which they actually made into a database, right? So what they did in this paper was they demonstrated HESs residing in protein coding genes are often tolerant of naturally occurring synonymous substitution at vowel positions in their target sites. So this is quite straightforward. They allow some mismatches to during when the protein binds to that DNA, right? At the vowel position is like G and U. They can interchange their positions, right? So these kind of mismatches were allowed. So they found it. Then they predicted a putative target sequence for newly discovered HESs. Okay. The result of this search were compiled in form of a database called home-based database. So basically, we'll be exploring this database and see what homing endonucleases are. I mean, how many homing nucleases are reported over there. Their structures are also available, and they have also given a searching tool where you can search for these sites in a given gene, right? So we'll talk everything with that aspect, but let's start. So this is one of the paper. Oh, I'm sorry. Home-based database is not functional anymore. I'm sorry. So many of the databases after a few years stopped working. So after that, there came a newer version of it, okay, which is LHEDES, and it is a homing endonuclease database itself that work like the same, and it was published a year later in 2012. It is an archive of LH, as LHEs. Initially, it was done only on LHEs, but later on they added every other family as well. But since in their paper they have mentioned only LHs, I have kept it here as LHs. So it is an archive of LHs that have been sufficiently characterized to be used as starting point for creating gene targeting proteins, right? So identifying combinations of endonuclease scaffolds and DNA targets that could be used for targeted gene modification. So this website actually had three major, you know, features, as you can see. So the first thing which they considered was enzyme and target site entry. They have a collection of, you know, these site entries and the enzyme responsible for it. We'll see. So then they have given each nucleotide position some weight values, right? Specificity profile entry or position weight matrix. We'll be discussing in detail about PWM. I'll show you how to calculate this, not using a software but by hand. How you, if you are given a sequence of suppose 7 length, how are you going to calculate the PWM using pen and paper, right? So the next thing is LH search. So anyway, these things will be discussed. Okay, let's start with the first one, enzyme entry and target site. As you can see, the CREI gene over here, they have a collection of genes. I'm just showing one of them. So this is the target sequence for CREI, right? And in blue, as you can see, GTGA. This is the central four. As I was saying, every homing endonuclease has this central four recognition side, as you can see, and from there will be a right hand side and the left hand side. In right, they, in convention, they give it as positive numbering, and in the left hand side they give it as negative numbering. This positive and negative are not magnitudal, it's just naming convention. Okay, it's not -1 +1, it doesn't have anything to do with the magnitude. Okay, these are just naming conventions. So in the database, you will also find one more thing, which is here, the 11 -11 -1. This is the contact information with which amino acid residue this sequence is actually, you know, interacting, right? So we have this DNA sequence, and we have a protein that goes and binds to it and cleaves it. So which are the places where it binds? So these, they have given. You can also find details about these things as well in that database. So let's just see what this whole enzyme and target site entry is about. It has an updatable collection of wild type and engineered LHs that have been showed to clip DNA sequences. Right. The target sites are listed as 22 base pair sequences. As you can see, there are 22 base pair sequences that exactly span the center of 4 consecutive bases. As I said, these 4 consecutive bases that are converted into manually cohesive 4-3 overhangs by all LHEs. Every LHE has these 4, and then they have these overhang flanking regions. Right. The left hand side, there is the -1, -11 to -1 is contacted by residues from N terminal. Right, the N terminal end, while the right hand side, the other high, the 1 to 11 is bound by C terminal. This is quite basic, right? Everyone understands this. Yeah, are there any questions? Any doubts till now? Okay. Anyway, moving forward. As I said, position weight matrix, right? So what this is the actually core of designing any tool. So what happens here? Let's start from the beginning. The specificity profile reported as PWMs that have been experimentally determined for a number of LHEs can be entered individually. Okay, so what happens? Suppose you have sequences. Suppose you have sequences which correspond to, you know, nucleus activity. So if you find the relatedness by multiple sequence alignment or anything, suppose you are doing multiple sequence alignment itself, so you will see that each position will not necessarily have A in it. Sometimes it may be T or G or something like that. So each position, what is the probability or what is the weight of finding any one nucleotide? That is what position weight matrix is. Like uh, it is represent, as you can see here, it is represented with these alphabets. Suppose here, as you can see, G is huge, right? Number 4, and T is small. So if you see here, G is 1, T is 0.8, and the remaining are 0, 0. That means in this place, the probability of G happening is 1. Not probability exactly. This is position weight matrix. This is the weight they have given. And you can, of course, change the weight according to what scores you are trying to give. But I'll show how to do this, calculation actually. So as you can see, for each nucleotide position over here from -11 to +11, right? For each position, what is ACGT, right? What is the probability of getting A, C, G. I'm saying probability again. It's a mistake. It's not it's not probability. It's the weight, right? So, like in suppose we go for number three position, you can see T is the highest number. It's one, right? You can see the T is marked as the longest one over here. Then we have G, C, A. So according to these values, this thing is marked up. So that is what is explained here. These matrices are displayed in graphical format as a sequence logo plot in tabular format, right? So these sequences will be there, and their occurrences are given by the length of that alphabet. The ability of the enzyme to cleave DNA target that contain a given nucleotide at a specific position within a target site is indicated by the letter height, right? So, what is the probability of finding that nucleotide in that position, right? So, they have used ability instead of probability anyway. Let's see how this thing is actually calculated. As you can see here, one there the position in it there is one, but then again it is 19, 13, and 45. Of course, it's not probability, right? If it were probability, then the summation of all this should have given one. Since this is not probability, you can see when you add all of them, it goes above one, right? So this is the reason it's not a probability score. So what is this PWM then? So trust me, this is the most easy thing to learn. This is the easiest slide that I'm going to explain here. Suppose you have uh, seven sequences, seven interrelated sequences. Suppose these are seven sequences, you know, are the targets for some endonuclease. Okay, some endonuclease has these seven targets. Okay, so now as you can see, the length of this sequence is 3, 4, 5, 6, 7, 8, right? Anyway, so what now we can see here at the first position, it is given as T, right? So there are four T's and one A. Sorry, four T's and three A, but still it's given as T. But that doesn't mean that if there is an A, it will be a mistake, right? So to accommodate A over there, we need we need to assign some weight to A as well, right? So although this is the consensus sequence, some weight should be given to A. So how to give that, weight to each sequence? Let's understand that. Suppose now from this whole matrix you want to convert into numbers, right? So how many A are there in position one? One, two, and three, right? So it is three. And how many T's are there? There are four. One, two, three, and four, right? So here we have three. Here we have four. In second position A, there is just one A, right? There are two C's, four G's, and so on. So these are the counts, the frequency counts in that particular position, what that nucleotide can be, right? So now what we can do is we can get how many total sites are there. There are seven, right? So out of seven, we are getting three A's. So 3 by 7 is 0.43. As simple as that, right? You have to just add all these sequences and the probability of getting three A's out of seven sequences. That is how it is, calculated. Same way here T. If you see, like four T's out of seven sequences, four of the times it was T, and three of the times it was A, right? So it is 3 by 7, then 4 by 7. As simple as that. If you do this calculation, then you will find this matrix over here, which will give you the frequency, right? 0.43 and 0.5. This actually is, when you add this thing, you will get 1. As you can say, the probability kind of thing. But it's not again probability, okay? It's a position specific frequency matrix. But still, if you add all this, you'll always get 1, right? So again, moving forward, this is just the formula how to calculate. I didn't show the formula because formulas are always daunting. People are scared. I mean, I'm scared of formulas. So first we understand how to do it, then we formulate it. So this is just what we did. The n I of j, that is one by one. Suppose we have three, right? Then summation of n r j, the whole number of sequence, that is seven. That is 4 by 7 for this t, and 3 by 7 for this. So this actually explains what the whole thing is going on, right? So I hope everyone is following me, right? Whatever I'm saying, or should I explain again? Okay. Anyway, moving forward, this is still not position weight matrix, right? So let's change, there's one final step to convert it into position matrix, right? So this is what it is. Position weight matrix is finally, calculated after you divide the whole thing by 0.525 in case of nucleotide, or 0.5 in case of amino acids, right? This is the prior probability. So what we do, we divide this whole thing, 0.43 divide by 0.25 and take the natural log, and that is the position weight matrix. As simple as that. There's nothing complicated, I hope. There's just one thing, you just have to calculate the number of sequences here, the frequencies, right? Get the counts, change it into frequency, then take the natural log of it. That's how you'll get the position weight matrix, right? Are there any questions? Actually, I'm expecting one question here. Okay. The question is, you cannot take the natural log of zero, right? Whenever there is zero, it will say invalid. So what are you going to do in those cases? So in those cases, what they do, they give a pseudo weight, right? A pseudo weight of 0.01. Suppose, then what happens? This numerator will not be zero anymore. It will be 0.25 or 0.1. 0.1 into p of I is 0.25. That is a 0.25 itself, of course. 0.25 and divide by 7. So the numerator is changed by giving some pseudo weight or something, so that we don't get invalid result, right? So this is the whole point of position weight matrix, right? So this is the last feature of LHE server, which is searching individual genes or a collection of genes for potential matches against LHE targets, right? So whenever you give a sequence, it will give you the potential targets and the score over here. What is the score of getting such a nucleotide position? Anyway, so from here, let's in the first part of the practical, I thought I would be showing, the usability of this LHE database. So this is the website here, homingendonuclease.net, right? If you go there, you'll find this database which I was talking about. Okay. So in this database, if you go to homing endonuclease browser, right, you'll find the target sequences along with the four central nuclear sequences, and these are the HEs, right? So these are the wild type HEs, and when you scroll down, you'll find the engineered home HEs that we just discussed. And then we have again chimeric home nucleases, which again we discussed, right? So we have discussed all three parts of it. So the thing is, when you click on any of it, suppose where is CREI? Okay, suppose we go to CREI over here. I just showed the screenshot. This is the same thing. Okay, so this is the target sequence, right? 11 to 20, 11 minus 11 to plus 11, right? So this is the FASTA sequence of that endonuclease. You can take it and make your crystal structure, or directly get your PDB file from here, where you can rotate it in PyMol. So whatever you do, the thing is, we are concentrating more on the sequence part, not the structural part. So in sequence, as you can see, ACGT, this thing, this is the PWM that I was talking about, right? So I hope you can understand it now, what these numbers show. So the point of explaining the whole concept was, many of the time people complain that they don't get results after using some bioinformatic tool, and they blame the bioinformatician. So the thing is, we have to know what we are using, right? What that tool is based upon. If we concentrate upon that, then only because that person made that software which was targeted on some different problem, which is not the same problem that you are facing. So first we have to make sure that this solution fits our problem. Then we can hypothesize that yes, this tool we use because we thought that since this tool solved that problem, we are very sure that this was going to solve our problem as well, because these are the similarities in our problem. So that's how we have to model, or ask the question. So the whole point of explaining that PWM was understanding this thing. Whenever you go to this website and try to blast it or search for any sequence, you should know what the suppose eighth position of ICRE one should be, seven should be, right? So in your case, in your target this might not be the same, but how much is allowed because of that PWM, right? We can calculate it from you. So that's the whole point. So these are the and this is only possible, the calculation of PWM is only possible because we already have a lot of sequences, reported sequences, right? Those bioinformatic analysis which were done initially, they reported these multiple sequence analysis and stuff like that, based on which they came up with the whole idea, right? So this is the uh, okay, so this is what the whole tool looks like. Then, as I said, there is a searching algorithm as well, and we have central for identity search PWM, and they have different algorithms. So let's go for just one central for search, right? Suppose we have a sequence like this, a gene sequence, and we want. Can you see it? Right? I hope you can see it. So, if we have a gene target like this, and we want to see if it has sites for ICRE, or ONUI. So, if we want these things, we can just confirm it by clicking search, right? So, for this, as they have showed, ICRE, the sequence that we entered, these are the places which has a similar sequence, right? And this is based upon that ESW. So GTGA, and in the wild type also there is GTGA. So this GTGA, just like that, they'll search for the whole thing and give you the result. Okay. So I think this everyone is clear how this thing works, how the search algorithm works. They'll search for the central for. Suppose I'm searching for CREI, then GTGA will be searching my sequence, and then each sequence to the left it will search, then each sequence to the right it will search. For each letter, they will check what the PWM is, and then finally give the score. I hope this is clear, right? If this is clear, then we'll go to the next part of our practical. This is a very short practical. Actually, this is just a demonstration of what there already is. So the next part is, suppose we don't have a database like this, right? We don't have any database, and we want to search for some. Okay, not this one. I'm sorry. Okay. Suppose we want to search for some novel targets which are not there, right? So keep this problem in your head while going forward. Select the best target, filter criteria. So first of all, in that is not the database that I showed is not like blast and all. Okay, it is just these are the criteria. This is the score criteria. So PWM is the main thing. That was the reason for explaining PWM in detail. So the criteria is this, right? Suppose in the consensus sequence, the 10th position, there is a high chances of getting a T. Okay, and in your case, you are getting an A. So this is how they will calculate the whole thing, right? The whole point of showing you this slide. Wait, I'll show you again. This slide. Wait. Was to make clear what that PWM is, right? That's what the scoring thing comes from, right? So let's say these are the homing endonuclease genes, different targets that has been reported. There are hundreds and thousands of reported targets. Suppose we have, right? Now that's a 22 base pair length of a target, right? So that will never be same. It's very hard to get such a long specific sequence, because as I said, 1 by 4 to the power 22 is very hard to get. The probability of getting 1 by 4 to the power 22, right? So so next it is PWM. That's what I'm saying. So what happens now is, after getting this thing, we are creating a matrix where we count the number of A, number of T's, and like that, and fill it up here. Okay. So A, 3A. So we have 3 over here. T, 4 T's. So 4 over here. Like that, a PWM is calculated, right? So that thing will be used. Okay, fine. Let me complete the whole thing. So after counting the thing, we are dividing it with the number of sequences. This 7, right? So 3 by 7, 4 by 7, 1 by 7, 2 by 7, 4 by 7 again. So like that. it goes on. I hope you're understanding. Okay. So after we get this, we are trying to find the natural oh, sorry. We find the natural log of this value divided by 0.25, which when you will get then you will get 0.54, right? 0.82. Now if you add 0.82 and 0.54, it's more than 1. So these are not probabilities. That's what I was saying, right? So as you can see, each sequence has a value associated with it, right? So the A in first position has a probability of getting an A here is 0.54. So, so if you see here, like A 1, 2, 3, 4, 5, 6, 7, 8, right? And here we have in A position 0.54, 0. Minus 0.58, like that. If you see, if you pay attention here, this is the same thing going on here, right? Same thing, right? The same thing here. So they calculated the whole thing and reported in their paper and created this database. They are not showing the consensus sequence. Of course, they are not going to show it. But this is the whole logic of calculating the thing, right? Tapashri ma'am, am I clear? I mean, am I able to explain it, or should I explain again? So in each position here, as we saw, in this case, we have 1, 2, 3, 4, 5, 6, 7, 8, right? Up to 8 we have here. And in this case, we have 11. 11 here, sorry, 22. 11 upwards, 11 downwards, right? So 22. The same thing that we saw over there is happening here. The same thing. Okay. I hope it is clear now. Okay. Moving on then. Now, suppose this is a very well defined database, right? We have everything, PWM and stuff like that. But when you are coming up with something novel that no one else has done and you won't get this PWM, the first thing you have to do is search for it, right? You just want to search if there that sequence is available in the gene or in the genome or whatever it is, right? So suppose you're trying to find something novel. You're someday you are working in your lab and you came up with some endonuclease which is cleaving here and there, and you just need to know the what how to get the targets for this endonuclease which is not there in any database. So how to work about that, how to you know go for that problem. Let's understand that. So first let's understand the problem itself. Okay. What was it? So let's keep this problem in our hand, and at the end of the session, we should be able to solve this problem. Actually, I wanted to show this problem at the end so that you know people can work on it. But I thought of showing this problem at the beginning so that this is there at the back of your head while we proceed now. So in a hypothetical situation, suppose you are given an eco R1 like nuclease enzyme. Okay, it is like eco R1, but it is not eco R1. So it has been discovered, suppose in your lab. This is a hypothetical situation. Okay, this doesn't happen with slight variation as follows. Right, the recognition site is GA, then in eco R1 it is GAA, right? But suppose in this case it can be anything. After GA, the third position can be taken by any of this nucleotide, right? Any of it. It can be ATGC, right? And then we have that TTC. That is the first variation. Then we have another condition here that we have a CPG island. Can I draw here? I think I can draw, right? Ah, great. How to erase it, by the way? Okay. Oh no. Okay, we have an eraser as well. Okay, so suppose there is a recognition site. This is the recognition site. Okay, there is in place of there is a difference here. It can be anything. In place of A in the third position, it can be anything. Okay. Now we have sequence to the right, right, and we have sequence to the left, right. So now what they are saying is we have in the upstream, right, some 5 to 10. It's not defined, it can be 5 or it can be 10. From between 5 to 10, again you don't know what exact it will be in it will depend upon the organism, right? So there is a variation. Suppose actually in biological problems, that's how you get your problem. There won't be exact 5 or 10 or like that. It's always a vague thing. So 5 to 10 signature is here, right? Suppose here we have 5 to 10, Okay, and then we have CPG island. That means CG like that. CG like that, right? This whole thing is GC. The same way we have some sequences here, which is from 5 to 10 length. We don't know what it is, right? And then we have Tata box, right? TA like that. Oh my god, my handwriting is so bad. So anyway, this is how the problem is. I hope you keep this problem in the back of your head while we solve, while we understand to go about the whole problem, right? So let's keep this problem in our mind and think of a way to solve this problem by understanding a different concept of searching. Okay. Suppose I ask you to search EcoR1 gene in EcoR1 target site in a given genome. What you can do? Ekdam simply, what you can do is Control F, right? And GAA-TTC, right? So those are the sites where it will go and bind. But in these cases where there are flexibilities, right? It's not very strict. There are flexibilities here, right? So Control F is a very strict search. We don't want a strict search. We want a flexible search for it, right? So the first thing I want to introduce is, okay. Oh my God, where is my eraser? Okay. So this is called regular expression. So regular expression is an ability to match a string of text in a very flexible and concise manner, right? So in this case, there are few rules. We'll see about the rules one by one, and we'll go to directly to the practical without wasting much time over here. Sir, am I over time?
It's okay. How much time you are going
So 20 minutes, 20 minutes, 15 to 20 minutes. Okay. So these are the rules. Let's start with the first one. So suppose anything has an A, B, C, D, anything, we have a question mark following it. That means we are essentially asking if there is an A or not, right? This is OR gate if you remember from your Boolean algebra. So zero or more of a star, which will give you, if there is no zero, then also give me a hit. If there are A's, then also give me a hit. It gives you everything actually, universal. So then we have one or more of A. If there is an A and A is continued by another A and another A and another A like that. So give me the whole sequence of A. That will be done by A plus. So just try. You don't need to memorize all these things. You just need to understand it. There, the whole list of arguments are given in the website. You can check. So anyway, the next thing is, suppose we want to see exactly three of it, right? There are three consecutive A's in a sequence. So then do this A in square brackets. It's three, and then three or more. Then we'll do a comma, right? So I hope you can get a brief hint of what can be done here. 5 to 10, right? This will be 5 comma 10. Anyway, we'll be solving it at the end of the whole thing. You should be able to solve it. That's the whole point. So the next thing, let's search. Are there any questions, please, before going forward? Because right now I'll be just discussing about the rules. Are there any questions? Should I explain anything again? No. All right. So the thing is, let's see the next. Okay, thank you so much. So then let's see the next rules here. So suppose I want A, B, or C in a given place, then I'll put it inside square brackets, right? So again, I think now you can understand how to go about this problem here. A, T, G, or C, right? Anyway, we'll be solving it anyways, but this is where you are going to get the solution. So a character which doesn't have ABC, right? Then we'll put this cap. Okay. A character in range of A to Z, but you won't be using it in biology. So these two can be, actually this concept comes from computer science. Not It's not a biology concept. Okay. This thing is a very old concept used in computer science for mining data. Suppose you have a jumble of data and you need to find patterns out of it. They use regular expression to extract the things. extracting emails or anything. I'll be showing the applicability of regular expression in a few minutes anyway. So, so suppose I want to now match any single character, that's a dot. It will match anything. Then suppose A or B, given one or the second, that is done by a pipe. Any white space, suppose you are given a letter and you want to know if there are spaces in it, just do a slash s. The same thing can be done for nucleotide sequences. Suppose there is a gap in your sequence for some typographic error, you can search that by going by typing slash s. So like that. Then start of a string is given by a cap, not inside the bracket, okay, just a cap. And then the end of a string is given by a dollar. All right. So I think we have seen some of the rules and we can just start working with it, right? We have some of the rules here. Let's start. Part two. Suppose now we want to search for poly A tail in a given sequence. I hope everyone knows what poly A tails are, right? So they are 250 nucleotide sequences, and the smallest one starts from 50 to 200. They have reported. So if we want to scale in a given sequence, we'll need, if there are stretches of 50A together, right? So that should give us the poly A tail. So how to do that? Let's start it. Okay. So for as I said, regular expression, there is a website called regex 101 where you can test your codes. Okay. So where you can test your codes, and they will give you the interface for all these languages as well. All right. So suppose okay, let's start with our sequence. I have a sequence over here. Paste this sequence and put some a at the end. Right. Suppose this is our poly a tail. Now we have to find such a way that this a over here should be captured by searching. Suppose we have some a over here as well, some a here as well. Now, the first thing that we can do is, right, remember the square brackets. If you have to, okay, these are quick references here, right? So suppose you want to go for, see, the single character A, B, or C that I showed there, it was taken from here, right? A, B, or C. So what we want here is A, right? But the problem is it is giving us hit every A it is getting. All the A's are reported back to us, right? So we don't want that. We want only the A which has at one stretch it is there. I mean, there are no other A's in between. In Tata box also there are many A's, right? So we want A's which are in a continuous stretch, right? So for that, if you remember the second rule, the plus, right? This rule, one or more of A, right? One or more of A. So if we give one or more of it, we are almost there, but still there is one problem. As you can see, the A over here, here, and here are also captured, and of course some A which are occurring here and there. Okay. So what can we do? We need to just report the last because poly A tail always occurs at the end, right? End of an mRNA. So to go for the end again, as I mentioned there in the rules, here this one, end of a string, that is a dollar. Just put a dollar here and you got your sequence, right? So this is how you can find a poly A tail, by using square bracket a plus and dollar. That will be the regular expression for finding a poly A tail in a given mRNA, right? So I hope you understood the basics, I mean how to go about it, right? So after getting the poly A tail, let's see the next one. This was the first one. I hope, I want some input from you so that I know people are there watching me. So let's do this together this time. So codons involved in synthesis of leucine and arginine. And suppose we are given a gene, suppose some collection of genes, and someone asked us to just find if there are codons which are going to synthesis leucine or arginine. So how are you going to do about it? How are you going to search? Right. First, we need to know what leucine the codon for leucine is. So if the codon for leucine is Cu, then at the last position there can be anything. This is a wobble, right? So this can be anything, A, U, G, C. Whatever Cu, A also goes for leucine, Cu, U also goes for leucine, like that. So now you need to catch all these codons which searches for leucine, right? So for your reference, I have given here Cu, U, Cu, everything will give for leucine, right? So how are we going to do about it? Okay. So suppose we have CUG, then some sequences, then again we have CUU, then again we have some sequences and CUG. Suppose let's, I have given CUU, CUG, right? So now we only want the sequences which can give an arginine in return, right? And leucine in return. I'm sorry. Okay, leucine. So it is Cu, that is, actually we don't even need to do this, Cu, and then here A, T, G, C. Oh, okay, I think we messed it up. U, G, C. As you can see, these three codons are now captured because Cu is taken as such, and anything which is coming inside this bracket has been captured. Like CUG is captured here, CUU is captured here, and CU again G. Okay, anyway, let me change it to suppose C A. It is getting captured, right? Like that. So, okay, can you give me for the next one arginine? Can anyone type it on the comment chat? I mean, I want some involvement. Otherwise, it's like I'm just speaking over here alone. Anyone wants to try? What should be for C, for arginine? CG, that is. Okay, I guess no one is listening. So it's CG, right? Yeah, thank you so much, Poonam Sen. But it's almost there, but the bracket should be square bracket. Okay, that is not a normal way. Thank you so much for being involved. Right. So, okay, as she showed us here, it is AUGC. Suppose I'm putting, it won't come because, yeah, exactly, the square brackets over here. It should be in square brackets, right? We already have some sequences over here having C, C, G, C. Anyway, thank you for your involvement. I have Bushra Khatun saying one by. Can you explain why you are using one as a denominator and dividing it? Since it's just screen manipulation. I mean, I may not know. I want to learn what it is. Why are you doing one by something? Bushra, ma'am. Anyway, just moving forward. I think people have understood the logic here, right? So let's go to the next problem. The thing is, you should not use FASTA file. That's what I'm going to show you. This is just for testing your regex 101 is just for testing your codes, if your regular expression works or not. But if you're going to deal with your data, suppose you have NSIC data or genome, it will never work on their server. You have to do it locally in your own terminal or using some programming language. So actually, regex is supported in every programming language, starting from Perl, Python, anything. So it's very easy to implement. I'll show by the end of the session. I'll show you how to add the Python, add regular expression inside a Python code, and again hosted in a web server. All right. So this is done. I think up to this one, it's done, right? So I'm giving three more. Can you quickly solve this? Suppose we have 25 or more base pairs of G without a G. I need a stretch of DNA where there is no G. At least 25 base pairs of a DNA stretch where there is no G. Can you write it on the chat? Anyone wants to try it? Okay. This is what you'll be using here. Okay, the square bracket. So what I need is something without a G, and it should be how much? 25, right? So it should be 25. So there is this, of course, this polyetail doesn't have a G. So we have, we can search for a G like that. If no one tried, okay, fine. So anyone wants to try the next one? Terminal triplet codon. It's almost like the poly A tail. Suppose I want to know what is the end codon. Is it a nonsense codon or not? So I want to check it. So I need the terminal triplet codon. Can anyone write the regex expression for finding the terminal triplet codon? The nonsense codon. I want to know if the given sequence is ending with a nonsense codon. So for that, can you write a expression? It's very easy. It's just like getting the poly A tail if you were listening. Okay, I think no one is there. So yeah. Okay, so okay, I'll leave these things. I'm moving forward then. I'm moving forward. I hope you people do it in your home if you want to. Of course, there is no hard and fast rule. So moving on, moving on, moving on, moving on. Now, using the same thing, we can search for target sequence of Cas9. DSA-Cas9 is a variant of Cas9 protein which has a PAM sequence of NNGRRT, where R can be either A or G, right? And followed by which is the PAM sequence, and before that it has a 20 nucleotide RNA guide RNA sequence. So does anyone want to try what the regular expression for this endonuclease should be? I think not. So anyway, the thing is, if you can find a regular expression that can search for targets like this, you can implement it in a web server like these people did, and they published it in it's a good journal, bioinformatics. So all they did was getting the regular expression, finding some score, and giving a you know response. So NNGRRT of a PAM sequence. If even if no one is watching, let me try. So the first thing should be AUGC, right? AUGC, which is of 20, right? We know guide RNAs are of 20 nucleotide sequence. Then we need NNGRRT, right? Then we need NNGRRT, right? This can be R is A or G, A or G, R. So this is the solution for getting you know, this nucleotide sequence. Anyway, moving forward. So the same way you can search for G quadruplexes as well, and the same thing these people did and reported their tool as QGRS mapper. They did nothing but just search for the G quadruplex signatures, right? So what they did was they took this expression, put inside a web server, and get the sequences. So let's see the last thing again, letting going back to our problem. I thought by now everyone will be able to solve this problem, but anyway, the thing is, suppose we want a recognition site of AAT, this thing, then it should be given here, right? This is the recognition site, and then we have CPG island of 5 to 10, which is given here. I mean 5 to 10 sequences in between. Then we have GCC sequence over here, data box over here. I thought of showing this everything, but we are running late, so let it be. So last thing that I want to show you, I'll take five. Sir, I'll take 5 more minutes. Is it
Yeah,
okay?
yeah.
So in this thing, what we can do is, I need it. Where is it? Okay. So suppose we have this thing. Uh, okay. s- where did I keep it up? I'm sorry. Okay. This is the folder, and I'm okay. Suppose we have folder here like this. Okay. These are the content of the folder: static, template, demo, image, and all that. Okay. Now what I want to do is get all these sequences. Get the regular expression over here inside my computer, so that every time I ask for something, it is able to give me a response, right? So the thing is, what I'll try to do is, whatever we did over here, see, this is for testing, right? So if you go code generator and look for Python, you'll see all the codes that are generated here. But these are just very gibberish. If you are not from computer science, it's very daunting. So I want you to understand what these things are without even discussing what these things are. So the thing is, we'll go here, right? And first, let me explain what a flask is. In Python, suppose whatever we were talking here, like getting a sequence, searching for the regular expression, and giving an output. So to get the whole thing in a streamlined process, we have a application called Flask in Python. Okay, what Flask does is it serves as a local server. It will get the input from user or from the browser and give a response to the browser. So it's very easy to implement. So what all we have to do is, we have to make the whole structure, the whole container in a Flask, and put that regular expression inside somewhere there, right? So the easy way to do it is just first, the first few lines are the dependencies. What are you going to use in your whole program, right? Then we are just saying I need Flask, right? So if you don't understand something, just post it, okay? So this is what the two lines are. Then moving forward, I need a home, right, where one should go and search for it. Suppose I have a home here, right? Okay. So I have home here and some few lines of code. This is actually called HTML, where I'll be getting the input files, right? So the text area will say where the
Text system
input.
visual, please zoom.
Uh-huh.
Text system visual, please zoom.
Can you see it now?
A bit more. Yes, okay, okay.
Okay. The thing is, the first few lines here are the dependencies, what you are going to use in your program. Okay. Then these two are declaring that you are using your Flask as the name of app. Okay. Then we have this is called home directory. You'll understand this as we run this program. Then this short thing over here, these are the HTML code which will be posted to the browser. So whenever I'm going for the home, I mean I'm asking for home through Flask, what Flask will do is show send this thing inside a web browser. Right now let's run this thing, so to show what actually it's doing. Wait, let me give a output as well, otherwise there will be an error. So where is it? The result. Yeah, right. So suppose I have a result here. I'll explain everything, don't worry. So what this route means, the place where your browser will go, right? Okay, once I'm there, I'll show you. Where was I there? Where was I? Okay. So the thing is, this is the home directory, right? The home page. In home page, you will find these HTML codes, and it will return something. I'll talk about what is going to return, but let's first see what a home page looks like in this, right? So let's start with our terminal, we go there, Python 3, and just type what we have here, demo.py, right? Demo.py. Hopefully you don't get any error. We are getting any error. Okay. Indent. All right. Maybe there is a problem in copying and pasting. Let me paste it properly here. So this is the home, right? This is app. Actually, I created this script beforehand so that I don't face any problems while showing. All right, just bear with me. Okay, so
You can
this is,
share the script with them. No
yeah,
problem.
it will be in GitHub, but they won't understand the script unless they.
Right.
So this is what it is, and let's start it now. Why is it not working? Indentation will. Okay, I'll do the one which I have already created. Okay, itchy.py. Python 3. My God. Python 3 itchy.py. Right. So when you do this, okay, let me explain in this file itself. So I have this home where I have HTML files, right? So let me go and search the home now. This is the link where it's running. The whole program is running on this local ID. Okay. All we need to do is go there, we go there, paste it, and we get a sequence. Just a normal layout. Okay, where we are going to enter the FASTA sequence. All right. Suppose any sequence we need to give, whatever we are doing here, I'm just replicating the whole thing. This place, this where we are entering the, I'm sorry, where you are writing commands, it's just a plain Python script. I'm not using any software or anything. It's a, oh, okay, fine. So the thing is, this is the layout where you will be searching your, where you will be entering your FASTA sequences, right? And we are now asking our computer to search for a specific problem. Which was the problem? If you remember, this was the problem, right? This was the whole problem that we are going to solve, as I said in the starting of the session. So to solve this problem, this is the regular expression. I didn't explain it because I thought people are bored over here. So anyway, I'm just taking the expression. I thought that we'd solve it together. But anyway, this is the whole expression. Okay. So what we need, whenever a gene sequence is entered, we should be able to tell the user that this thing is there, if this thing is there or not in their sequence. That's it. Okay. If there is a novel endonuclease which has a recognition site like this, which has CPG island before it, which is a Tata box after it, and you don't have any tool to search for such signatures, how are you going to do it from the scratch by your own? That is the whole, that is the whole point of the whole of this session, right? To be able to search for recognition site or target sites from the scratch. If you don't have any resources, how are you going to do about it? So that is the whole thing. Okay. So as I said, our program is over here, right? Where is it? Where is it? Oh, yeah, this thing, right? So let's enter a sequence. Enter a sequence. I know this sequence has that CPG island Tata box. I just kept it so that I could use it later on. So suppose I enter it. Okay. Now go to submit. It'll give me the matches. There are two sequences of that signature. See CG, that's CPG island, right? Then this thing is over here, right? Now this is the main part. The G, if you see here, GAATTC is normal eco R1, right? This is GAGTTC. Still, it's able to detect it because we are using regular expression, right? Then we have again a 5 nucleotide space and then again Tata box. This is the first site where you can, suppose you have found a new endonuclease, then you know that this site is the first one. And how do we know from where to where that matches? That is from 14 to 16. That means the sequence that we entered from 14 to 16 line, this whole match comes up. Okay, the range. The same way for the next signature, this CGCCG, this thing comes from 92 to 129, right? So actually now we have developed something where we can give the input and the computer will understand and see if that sequence is over there and give us an output, right? TK Inter module is different. I'm glad that someone brought it up. TK Inter and Flask are different. Flask is more like Django, if you have heard about it. Flask is not just for web development. I mean, sorry, app development. It can handle all the requests from suppose you're working with Nginx. Okay, when you are going for development mode, this is just running a software inside my local computer. But suppose I need to make a public, you know, domain IP where you people can also go and search. In that cases, I need to redirect the whole thing using Nginx server. I mean, Nginx is a program that will get the request and send it to Flask. So that whole bridging is done with the help of Nginx. And again, you cannot run Flask directly in remote. So for that, you need to use Gunicorn. This is called Gunicorn. So you have to use Gunicorn along with supervisor, right? These two you have to use along with Flask to be able to establish your website inside an Nginx server. Anyway, the thing is, this is the sequence, right? Okay, so you might be asking, why did we go for so much of trouble? We could have just used a terminal to get these things. Of course, we could have gone for a terminal, but using a terminal is a bit difficult, these cases. So it's better if you have a user interface, because all the websites that you see nowadays have a very well defined user interface where people can give their input, get the output, and stuff like that. So this is the whole thing that I was trying to explain. But let's see what the result here is doing. Okay, since I explained this thing, this is the form, which is the layout, and this is the response. That means whatever regular expression that we entered, it is here. There is re.findIter. Okay, this is the module, re module that I'm using from Python. All right. Then inside this re, I am giving this regular expression. I hope you are now familiar with an regular expression. Just by seeing, you can know that this is a regular expression, right? So this regular expression is given inside this Python program, okay? And then I'm getting the matches using this. I'm running actually through a for loop here, okay? For loop inside the whole find thing. Anyway, these are not very important. One day you sit in front of a computer, you'll be able to do it. So these three lines of code are actually calculating everything, getting the input, seeing the signatures and giving a return value. So this is what is given as results. That's why you are seeing this thing result over here, result, right? This is the content of result that you are seeing over here. That is the whole point, right? So this is what a backend look like, right? This is actually a backend, and a front end is what you see in front of your browser. Okay, although Python is so powerful that your front end code can also be accommodated inside your back end codes, but it is not recommended. It's never recommended actually. So what generally people do is they make a different file like here .html where all the things will be there. Okay, suppose I open this home.html. I have a template. These templates are available in Google. You don't have to write anything from scratch. Okay, you just have to paste whatever you want to, I mean the part of your code inside these blocks. So what I did, I used these templates to transfer all the codes you just saw here, right? You saw these codes, these are in very crude form. People won't understand, right, what is going on. So what I did, I just gave a better layout. That is, I work with the front end. Okay, that is the difference between front end and back end. That's what I'm trying to show you. So what is a front end now then? When I changed everything, okay, and suppose I go to home now. This is a full-fledged website that is working on. All right. So whatever we learned till now, I have, given everything inside that code and made a tool which could perform this thing. That was our problem, right? The problem in hand, the first problem that we discussed, that it should have a recognition site like this, a CPG island like this, and this one like this, right? So now if we enter the sequence that we know have this signature, suppose we enter it here and we say predict if it's there, if these sequences are there, this if it satisfies. So if you click it, you can see target match. Yes, this is the target match which is there in the gene sequence which we entered is this. The target match is this. The range is this. Okay, that is all. So this is a fully working website with proper back end and front end and everything. But it is not in production design. Okay, it's just for development. For production, you have to go for engineers. That's a different story. We are, and it's out of scope for this talk. So this is how tools are developed. Okay, whenever you see, I am in I'm more into development. I don't do analysis. I am more into development of tools. So that's how you develop tools to make life easier for biologists. So I think with this I'll end my lecture. No, actually, I thought since I talked so much about regular expression, and regular expression is not just related to biology. It can be applied to many of the things. I mean, most of the things that are extracting of pattern. So that's what we do. We use regular expression. So okay, I'm not promoting my website, but this is something I did with the help of regular expression. Okay, I want to show it. I'm not very sure if it's there anymore. Um, no, it's done. It's done. It's done. Just
Maybe
yeah.
one few questions.
Yeah, you can ask questions for now.
So yeah, in the meantime, you can ask a few
any
questions.
doubts
Already
you
many
have.
questions has been there, right? Maybe we'll be able to take up few
Yeah.
questions.
Okay, so this is what I wanted to show. I created this website long back during the COVID pandemic when we had nothing to do. So what I'm doing here is I'm just using regular expression to grasp all the things that are available in our databases. But suppose we have these are data, active cases, recovered cases, like that. Okay, Sonitpur. These are districts of Assam actually. So you can see how many recovered and how many active, things like that. And here you have recovery rate. Oh, sorry, like this. But everything here you are seeing is done using regular expression. That's why I was saying the importance of regular expression. It's not just related to biology. You can do variety of things. The same way here you can see the status window. Suppose you want to see the recovered ones here, the number of vaccinated ones. So all these things are actually parsed through regular expression. That's why I'm showing it here. Like if you go to these websites here, okay, if you go to these websites, you will find this data. What I did was I collected everything from that website. I found the pattern when it said, vaccination done for Karnataka, Jharkhand, like that. For each and every one, I collected, and these are dynamically collected actually, and that's how they are displayed. So this is just to show you the applicability of regular expression. Regular expression are not just limited to biology or biosciences or anything. It's just a normal concept in computer science, which is used widely. So with this, I end my lecture. Thank you so much.
Yeah. So if there are any questions, please kindly ask. Or if there are no any questions, you can also write down no. You can just write down N.
I don't think anyone is there.
For your presentation. Yeah. Are there any questions?
It seems, I think it's
Yeah, there is
long.
one question.
One
Sir,
question to me.
does regular expression show that our search expression is correct? Uh, that's why we have websites like this where you go and test your codes first. Okay. once you test your code, you implement it, and then you see there are other things, other errors. For example, I'm telling you, I'm already much ahead of my time, but since this is a good question,
No problem. Please go ahead.
this
Please
is a
answer.
good question actually. How
Please
are we
answer.
going to know that our code is correct, right? Our regular expression is correct. Suppose I ask you, someone is trying to find Tata box and he writes Tata box like this. It's a mistake actually. Don't do this. This is wrong. The Tata box here are captured, but these Tata box are captured because it is capturing anything that is A or T, right? So after getting your output, you have to check, you have to verify where you are doing the mistake. And the best way is, if you don't know, if you are unable to find a pattern, if you are unable to find a regex, what you can do here is save your regex here and post your question. They'll reply you within half an hour or something. So if you're unable to do your reject, it's very complicated. Suppose it's a very complicated scenario, and being a biologist, it's sometimes difficult for us to think in that way. So what we can do here is we can post our questions where those people who are actually working in computation for like years, they can solve those problems in. What is the next question? Can we also translate? Translating ATGC to AA colon. Yeah, of course you can do it, but for that you need a bit of more coding. That is, first you have to get your sequence, then divide the whole sequence into 3 codons, right? You have to save it in a list, and then for each list you have to match it with the amino acid sequence which it corresponds to. I mean, it will start from regular expression, through regular expression it will go, and somehow it will end in database search, not even database search, list searching. That's how it will be done. Not just regular expression will be a part of it. It will be the starting point. That I forgot to show you, but in many of the papers they have mentioned the starting point of any development of any tool is regular expression. That's how they capture the whole thing. Then they process in their server. They will give scores and stuff like that. So for that you will need all those things. So that's a good question. ATGC to amino acid sequences.
Yeah. Okay. I think we have taken a very long time.
I'm sorry.
But I think it was a very useful session for all of you. I thought giving some, I mean, it is not very easy to do something of practical nature online with the challenges, but we look forward. So probably in future, if we have more such programs, we can do a little bit of more practical, and then by the time probably you will also be a little bit more skilled. Yeah. So let me thank Adhiraj for providing me the whole technical support, not only in conducting this practical part of this lecture, but also in hosting, and he has been in communication with all of you through WhatsApp. And then also Pulak is there. Pulak, please come over, and I would like to grab a frame with you. Come over, come over, come over. So please sit down a little bit. Yeah. So he is Pulak. He is also helping me, and probably all of you know him. He is also messaging all of you all the time. Of course, I cannot thank myself because I am the host.
Thank you, sir.
And I thank both of them. Right. So now, with this session ends here itself.